Just a little bit of discussion about early health economic modelling as a paradigm shift. How cost effectiveness models can look beyond clinical efficacy, which is, I guess, what most of you people would be focusing on. The process of building an early economic model. And then I'll cover a case study of integrated early economic modelling and a forecasting and valuation, and the example I'll be using is of a gene therapy. So a bit of myself, I've got a scientific background. My BSc decades ago, as you can guess, was in molecular biology. I did a PhD in oncology at the University of Cambridge. I spent several years working in Accenture's pharmaceutical practice in London. Actually, a lot of it was spent coding at the UK regulatory agency, the MHRA. I worked for a Cambridge-based biotechnology company that had an early clinical phase asset, and I was business development manager there. So I supported the out-licensing of platform technologies and early assets, and that included a lot of generation of asset valuation models in Excel to support negotiations. And I also sat on the target product profile team to help guide some of the design of the clinical studies. After that, I switched sides. I worked for a pharmaceutical company, again as business development manager, and in that role I was responsible for in-licensing specialty hospital products, and again that included assessing and valuing some of those candidates, as well as conducting some valuations for M&A purposes, and actually the main pharma did eventually get sold to Pfizer, so I was involved in that. I got a postgraduate certificate in international pricing reimbursement, and that's at Sheffield, partly down to frustration of when I was doing all that in-licensing and that valuation work of how do I allocate a price to some of these products, particularly when there was something which had no analogue for which I could sort of look for a reference price. I joined Cogenti as a health economist in 2016. I did a master's in economic evaluation and healthcare while I was there. And I've supported multiple pricing and reimbursement applications for pharmaceutical products, for medical devices, diagnostics, and that includes rare diseases, CAR-Ts, gene therapies, name it, and I've probably done it at some stage, so there's quite a lot of experience there. Have to tell you about Cogentia. So we're a little boutique consultancy. We're based in Cambridge in the UK. We work in a variety of projects ranging from assessing the value of early stage discoveries all the way through to bringing products to market. So we do a lot of work with submitting reimbursement dossiers to the National Institute for Clinical Excellence in the UK, but also some other agencies across Europe. The sort of work we do that's more relevant to this particular seminar, as you saw, we've got quite a broad range of activities for earlier stage products, things like landscape assessment. I won't go into all the detail here because we'll circulate the slides afterwards. A lot of primary research, speaking to clinicians and payers, and then obviously the early modelling, which is what I'll be talking to you about now. We have quite a large range of clients because we're quite a small consultancy. We really do have clients across the range. So you'll see on the sort of right hand side, I'll try and move my cursor without changing the slides, the University of Cambridge Enterprise, so those very early stage, just coming out academia assets all the way up to your AstraZenecas and your Takedas. So we work with all those sort of types of companies. Right, so going into early cost effectiveness modelling itself, and what we're talking about here is a real paradigm shift. Why am I talking to you about this? Well, as I said, having worked in sort of business development, I've heard a lot of these quotes in my time. This is a huge indication, huge market. We only did a tiny piece of the pie, so it's really worth going for this. Another very common one is that what's a gene therapy? We can charge $1.5 million per patient. Another classic one is a rare disease. People think because it's a rare disease, they can immediately charge $500,000 a year for a patient. These are all completely the wrong way to go about it. They are inaccurate. They give you a completely wrong idea of a product's value. At the moment, health economic modelling really sits, I guess for most companies, imagine that it sits somewhere around late phase 3, in that little phase when you're preparing for launch or perhaps preparing for health technology assessment. But actually what I'm going to talk to you about is that it really belongs a lot earlier on, and we've even had some clients with preclinical products who've come to us to do early economic models. Some of these applications include target indication selection, You can assess the potential price of a therapy, particularly where no analog products exist or where there's no active comparator. A lot of diseases have no disease-modifying drug on the market. There's just standard of care treatment with cheap generics just to manage symptoms. And obviously your budget impact is going to be massive. So you have to try and find some way of saying, well, what is the value of this product for payers? A second aspect is out licensing. It's a really good way of articulating your product value story. A lot of people have a very vague story. Yes, this product will do this or that, but it's a vague story. You need to give people some tangible ways of how this product's going to generate value. And this is particularly true where you're looking at product that just affects some type of biomarker, and often these sort of rare disease products might affect an enzyme. It might be an enzyme replacement therapy. You're looking at a surrogate endpoint. You're not going to get any hard clinical endpoints out of any of your clinical studies. So this gives you an idea of how do you get from those surrogate endpoints to these hard clinical endpoints, and what would the value of that be. In terms of in-licensing and acquisition, this really helps in terms of the due diligence process. We've had some clients who've actually asked us to do a quick and dirty early economic model because they're evaluating some in-licensing possibilities, and they want to know if they're goers or not and what are the risks, particularly if they think, well, is this something which might be risky for regulatory approval and particularly for reimbursement? Is a payer going to see this product and say, well, no way, you haven't got the evidence there, I'm really not interested. And then the final bit is in terms of planning for clinical development and also real-world evidence, because this really helps you identify which elements of your target product profile are of the highest value and really require the most evidence around them. You don't have to have evidence for everything, but you really need to know where to choose which studies you're going to do and what you're going to gather evidence for. So that does really help you to do appropriate planning of trial protocols and ensuring the right endpoints are included in your study. And if there's stuff that can't be collected in your clinical trial, could you do some type of real-world study, look at a registry that's going to help support that evidence. And it also helps you set some minimum efficacy requirements to achieve a particular price point in your clinical studies. Just pause, have a sip. So I don't know how much any of you knows about cost-effectiveness analysis, but what it does is it just captures costs and health effects associated with your new intervention and any relevant comparators, and it measures those health effects both in terms of quantity, so the number of life years that a patient will spend alive, and also quality of life, which is QOL for short. And quality of life is normally expressed on a scale of 0, which is death, or 1 to perfect health. Though you do get some states which are worse than death in health economics, where you can have some negative utility values. And those life years, the quantitative element and the qualitative element, quality of life, they're combined into a single measure of health, which is called quality adjusted life years, the famous QALY, which you'll have heard of. And this is essentially the product of the life years and the quality of life. And this is just depicted here on this chart. So you can see on the y-axis is the quality of life measure, and on the x-axis you've got your life years, and this line here and the sort of dark blue beneath it would represent your current standard of care. And you can see here that then the line above here will be your new intervention. And you can see that over time, for each year, there's a delta between the quality of life of your new interventions. I'm going to try and find my cursor again. A delta between the quality of life for patients on your new intervention versus the current standard of care. And similarly, you've got a delta in the number of years alive. And it's essentially this area between those two curves that gives you your QALYs gained. And that's simply how it works in terms of cost effectiveness model. And I think the real advantage of QALYs as a measure of health effects is that you're standardizing it. So you're using a universal metric that allows comparison between assets in completely different indications. And the second benefit is that it gives you a more quantitative means of measuring clinical value of a new asset rather than, clinician or payer interviews. I think companies often conduct these clinician payer interviews and they say, oh yes, we like this, and well, we don't like that. That doesn't really give you a quantitative way of measuring value. So we've talked about the sort of life years and the quality of life that gives you the QALY. And essentially a cost effectiveness analysis captures the total costs and the total QALYs for the intervention versus the comparator over a defined period called a time horizon. This is kind of synonymous with a forecast period in accountancy. So you might be looking at a 20-year forecast, a 3-year forecast. In health economics, we refer to that as a time horizon. And you're looking at your incremental costs and your incremental QALYs of your product over this time horizon, and those two metrics are then combined into a single metric, which is called the incremental cost per QALY, or incremental cost effectiveness ratio, the ICER, the famous ICER, which you'll have heard of, and that's depicted down here. So it's simply the sum of your incremental costs over the time horizon divided by the incremental QALYs. And just an important thing to note is that usually all relevant costs and QALYs are captured, not just the treatment costs. We often get clients looking at the total costs for the intervention and say, oh, what's going on here? It's not only the drug costs, but it's the cost of the administration of the drug, it's any healthcare resource use associated with managing the patient. So everything is thrown in, and that depends on the perspective. We normally use the payer perspective or the healthcare perspective. So in the US, there's a body called the Institute for Clinical and Economic Review, ICER for short. Very unfortunate. I don't know if they were trying to be clever, but yeah, it makes things very awkward. But ICER Group looks at the ICERs of new products and considers whether they are cost effective. And that is whether the ICERs generated are below their so-called willingness to pay threshold. And for non-orphan products, ICER normally considers ICERs up to $150,000 per QALY to represent a cost-effective product, but for orphan drugs that goes up to around about 500,000. And if you go onto ICER's website, you can normally find guidance explaining this. So the general rule is to be cost-effective is that your ICER has to be less than whoever payer you're dealing with, ICER, NICE, whatever reimbursement agency, they'll have a threshold, their willingness to pay threshold for a QALY. We often have people say to us, oh, we're the US, or 80% of my profits are going to come from the US. They really don't care about cost effectiveness. It's not a cost effectiveness market. Might have been true. I think that's probably changing. This is just a snapshot from a fairly recent publication in 2023, some research done by Tufts Medical Center. And they looked at some US commercial health plans, and they looked to see whether they were following ICER Group's guidance at all. What I've not put here is that the guidance was not mentioned very often in these reimbursement decisions. But when they looked at all this data, they did find an association between ICER's value assessments and how US commercial health plans covered specialty drugs. And plans tended to impose fewer coverage restrictions on drugs that ICER estimated to have more favourable cost-effectiveness ratios. I think the US does pay attention to cost-effectiveness, even if it doesn't necessarily say that it does. So one real benefit of cost-effectiveness models is that they can look beyond clinical efficacy. Obviously, you might be used to designing clinical studies, and you're thinking relative efficacy. Developers, as I said, they usually think about their new assets in terms of relative efficacy, in terms of, reduces risk of death by 50%, reduces incidence of seizures by 90%. And if these effect sizes you think are going to be large, then you think, oh, great, we can charge a really high price because we're going to have a really good hazard ratio or relative risk. But to payers, it's the absolute effects that matter. And a cost-effectiveness analysis helps you to see that value from their point of view. And I've got some examples here. These are real examples that I have worked on in consultancy. I've changed them a little bit so it's not obvious what it is. But on the left-hand side, you've got the developer's way of thinking of it, and the right-hand side, the payer's mindset. So one is here, my product allows all patients to have a stem cell transplant instead of only 5% typically, which is the only way to be cured of this cancer. But if you're a payer, you'll be thinking, well, a stem cell transplant costs me $100,000 per patient, and that's before I've even added in your drug costs. Your cure rate from this new product is only 10%. So I've wasted $100,000 plus your treatment costs on 90% of my eligible patients. Second example, my product reduces the risk of seizures by 90%, obviously reducing hospital admissions and death. But payer thinks, well, only a small subset of my patients have frequent seizures. Why would I pay this much to prevent seizures in these other patients? And then the last example is, my product increases survival from stroke by 50%. Payer, well, 50% of the additional survivors have serious long-term disabilities. So why should I pay for a product that just keeps people with poor quality of life alive at very high cost? So it's a very different way of looking at things. And sort of tied into that is extrapolation. This is one thing which economic models are really good at doing. So firstly, clinical trials are normally of a very, very short duration. And they might only capture, as I said, surrogates or intermediate outcomes of interest. A cancer study may only follow patients up until 50% of them are dead, so you have no median survival data available. Or a study in trauma or stroke or sepsis may only capture the initial period post-discharge, so perhaps the first 3 months. You don't know what happens down the line. If you've got a study of a treatment for a genetic disease, it may only capture changes in biomarker levels, or if you've got a diagnostic test, it'll only show you, your studies you've conducted only tell you the predictive ability of that test. It doesn't show you how using that test will then change the outcomes of those patients. And a cost-effectiveness analysis essentially captures costs and qualities over a time horizon that is usually far longer than the clinical trial period, and it also captures all clinically relevant outcomes, so not just the endpoints that you might be capturing in your study. It looks at that endpoints outside of that are often far more clinically relevant. And so often the way we do this is we link outcomes observed in the trial to other outcomes that may occur years later or may be considered clinically relevant and often more costly using these extrapolation methods. I've got a couple of examples here to show you of what that means. Survival data. Often these follow-up times in a cancer trial are usually not long enough to capture all death or progression events. But the survival after the trial observation period often contributes more life years and hence qualities than the observed time periods. You can see on this diagram to the right-hand side, the area under that curve, if you sort of moved it along until all patients were dead, it's actually going to end up being bigger than all the area on the left. And this is particularly important when you have curative therapies such as CAR-Ts, where there may be long-term survival gains. So if we go to the end of of this curve here, if you imagine this might be a curative product, it might plateau somewhere around here, and you're going to have a huge long tail of all these long-term survivors. That's not going to be captured in your clinical study. So you need to be able to extrapolate in your model to try and capture all that gain and the costs associated with those patients. And what cost-effectiveness analysis does is it uses some mathematical methods to fit what we call parametric survival curves. So you can see here, this is your typical Kaplan-Meier curve that comes out of an oncology study. We fit these smoothed curves using mathematical methods to extend that curve. Okay. So here's another example. We've got a rare disease with motor function decline. The clinical trial captures a measure called the gross motor function measure, two domains of which is the D domain, which is standing, and domain E, which is walking, running, and jumping. This is a continuous measure, so it can be anywhere between a range of values. How do you link that to costs or QALYs? So the first thing we can do is we can define discrete motor deficit health states that can be linked to costs and QALYs using the literature. So for example, we've got the gross motor function classification system, which is used in cerebral palsy. There's a lot of literature published on the costs associated with those different health states, and there's also quality of life values published. A second example is, it's a sort of a similar measure, the gross motor function classification in metachromatic leukodystrophy. This is a rare disease. There was a gene therapy approved for this sort of in the last couple of years, the GMFC MLD. Again, if you go into the public domain, you'll find data on the healthcare resource use costs associated with each of those health states and also quality of life values. So one thing you can then do, you can use either registry data or trial data to look at the relationship between GMFM-DE score and age. And that gives you an idea, well, over time, how quickly does GMFM-DE score reduce for each year of age? So an example here, say it declines by 5 points per year. And then using some predefined cutoff values, you can look at how quickly a patient moves to a worse GMFC category based on these cutoff values. So based on that, well, for me to move from GMFCS1 to GMFCS2, if I've defined a cutoff value of 75%, I know it'll take me 5 years to get there. And similarly, it'll take me 10 years to get to a 50% reduction, etc., etc., etc. You can then put that in your model. That is your progression rate on standard of care, and then you add in a treatment effect from your treatment. A third example is a rare disease which has an enzyme replacement therapy, so urea cycle disorder. These patients have defective arginine metabolism, and so they're at risk of having ultra-high ammonia levels, which can lead to hyperammonemia events that can result in hospital admission and death. And what we needed to do here was to model the risk of mortality from hyperammonemia on current standard of care based on data from observational studies, bearing in mind that this is an ultra-rare disease. So we had the observed rates of hyperammonemia of patients with the condition on current standard of care. We knew what their peak ammonia levels were on admission. Not enough deaths to draw any type of conclusion from that, but in the literature, we did have a paper which told you what the mortality rate was in different age cohorts and given different peak ammonia levels. So that allowed us to then work out, well, what's the likely risk of mortality for patients on current standard of care. You just then say, well, with my treatment, I'm going to have a certain amount of treatment effect. I'm going to apply it to the risk of having a hyperammonemia event, and then your reduction in mortality drops out of that, as well as a reduction in your peak ammonia levels. And then here's another one which I've worked on as well quite recently. This is a curative treatment that reduces vaso-occlusive events, VOX for short. So there was a trial in sickle cell disease, which captured as its endpoint a reduction in VOX. There was no comparator arm for this study. We know that people with sickle cell disease go on to develop a whole host of comorbidities as well as early mortality. So how do you capture this in your model? But again, using some published observational data, you can link the frequency of VOX with the development of multiple comorbidities as well as mortality rates. So I've just got a screenshot here from a published paper which showed that patients who had a follow-up VOC had a 56 or 55% higher risk of death than those without a follow-up VOC, and they also did this for a bunch of other outcomes. So essentially, just by reducing the frequency of VOX, you can demonstrate through these methods of extrapolation that you can also have an impact on comorbidities and on mortality. So in terms of
considering that the N though is really small, right? These are very rare diseases, so the N of patients must be really
yes.
small. How accurate can be the modeling?
It's you're right, it's very inaccurate, but when we work in health economics, we always capture the uncertainty associated with that. So I'm not going to go into details about all the methods of doing that, but later on I'll show you an example of how we try and demonstrate that uncertainty. But yeah, with rare diseases that is often the case. But normally reimbursement bodies are aware of that, and they tend to have often sort of processes that are tailored specifically towards rare diseases. So this is just a schematic of the general health economic modelling process. The first phase is conceptualization, and this is normally framed around a PICO. So the PICO stands for Population Intervention Comparators Outcomes. The first thing to say about population is populations are often not homogeneous. You might want to be considering some subgroups, because often subgroups can have very different cost effectiveness and which can lead to very different pricing. So some subgroups, an example might be pediatric versus adult onset, moderate or severe, different genetic mutations, etc. Your intervention is obviously your drug, your medical device, whatever. Comparators, this does not have to be the comparator in your clinical study. Often for FDA submissions, you often have to do a placebo-controlled study, but obviously in the real world, there might be a whole host of other comparators. You obviously can't have all these comparators in your clinical trial. So in health economics, we can compare between different comparators using a method called indirect treatment comparison, which is far too complex to go through in this session. And then in terms of the outcomes, obviously I mentioned we look at costs, we look at quality of life, we look at mortality, we look at the ISA, but we also try and capture disease-specific outcomes. So if we can, we'll get the model to churn out event rates as well, because this is very helpful for clinicians to look at and also payers to understand, well, yeah, I get it's changing costs and qualities, but what about the actual clinical events which I'm looking at? So we will also configure the model to generate these. So in terms of the conceptualization, the first phase is normally literature searching and clinician interviews. And we always try and design the model around some core health states, and these normally comprise the really key indicator of progression that can be linked to disease management costs and quality of life. So these could be liver fibrosis stage, it could be the stage of a cancer, it could be this gross motor function score, it could be responder versus non-responder. It's whatever is clinically relevant. On top of that, there may be other events that you want to include in the model that could either be conditional on being in a particular health state or completely independent of it. So this could be acute events such as these vaso-occlusive events, seizures, cardiovascular events, or they can be chronic events that clock up over time, such as cognitive decline. Lots of these neurodegenerative disorders will have a motor deficit element, but also a cognitive deficit element. Having done the conceptualization, which is normally done independent of the evidence there, it's much more designed to model the disease process and how your product is going to be altering that disease process. Then you go and try and look for some evidence and see, well, how can I populate this model with evidence? And in some cases, we do use phase 2 data. If it's a company that is just getting to the end of, has just finished phase 2 and is moving to phase 3, we can populate it with some early phase 2 data, or sometimes they might have done a burden of illness study, or they might have looked at a registry study. We can use that. Otherwise, in some cases, particularly in rare diseases, there might be nothing. And then we try and look at proxy conditions. So we've done that quite a lot recently, where we've had a new rare disease. We thought, oh, well, actually, the symptoms are very similar to this other disease, which already has a treatment. We just know that the progression rates are a bit different. But that's fine. In a model, you can do that. You can change the progression rate in the model, but actually have all the same outcomes as another disease. Having done that conceptualization process, we normally agree with the client. We then go on to build it. This is normally done in Excel, just simply because then it's really easy to adapt down the line by anybody. We also do some models in R-Shiny. Later on, the case study I'll be showing you is in an R-Shiny model, just because it's just easier to look at visually. The model usually includes a dashboard where you can configure key important assumptions, combined with a results table, because what you don't want to be doing is changing an assumption, flicking back to a different tab and going, oh, and try to work out what's going on. So we put everything on the same sheet. And we normally then also include a pricing tool, so that you can then, if you change a certain assumption regarding a clinical assumption, you can then run your pricing tool and say, okay, well, that's the impact on the potential price that I can get. So when you've done the modeling in Excel, this is often a really key go, no-go decision point about whether the potential price will really support further development in this indication. That's because we then use the price as an input in a discounted valuation model. And as I said, later on, you'll see how these two can interact with each other. This is just an example of a model dashboard, just to add a little bit of color. The top table, and I'll add that obviously this is anonymized, but everything's been changed, so you can't work out what it is. The top table shows the cost effectiveness results. I've lost my cursor again. The next table down, this is just showing you an example of the pricing analysis tool. And so you'll see on this slide, I mentioned before that different payers might have different willingness to pay thresholds for a QALY. So when you're doing your pricing analysis, you always have to try and work out, well, if my threshold is 500,000, obviously you can normally get a higher price if their willingness to pay threshold is higher. But it may not be that, because some payers have different thresholds even the same payer might have a different threshold for different products. They just, in some instances, they think this product is more valuable than that product. So I'm willing to pay more for a QALY for this product than I will for another. So this is quite helpful to have. And then the lower section, these are all the inputs values, so the model settings. And us as a company, we normally have a default value, which we call a base case value. And then we'll always be able to override that by something different, and then we'll have the value that's active in the model. And this allows you to do a lot of sensitivity analyses. So base case, I'm going to override that. Well, what's the impact on my ISA? What's the impact on my pricing? And just to give you an idea of the sort of input, so this section here is the baseline characteristics section, which can have quite a large impact on cost effectiveness results. Not normally gender, but certainly the starting age at baseline, because the time horizon over which you're accruing costs and QALYs can make an enormous difference. Pediatric indications often are hugely cost effective because you've got a huge time horizon ahead during which you can increase life years and increase quality of life. Here where it says type 1, type 2, this is an example of subtyping that I spoke to you about before. And then the bottom section here, this is where you're putting in all your treatment relative things. So the treatment cost per patient per year, the anticipated treatment effect. Here we've got a 90% reduction in progression in here. The stopping rule. So in some cases, for example, why would you treat a patient who's got to the worst health state if you're wheelchair bound, the product's clearly not been working for that patient, you might as well take them off. So we include things like stopping rules, discontinuation rates for treatments which are taken chronically, number of administrations per year. So that's just a small example of some of the inputs. As part of the model, we'll also do a one-way sensitivity analysis. So you mentioned the uncertainty, and obviously you put in your input parameters. None of these are a given, right? You don't really know when you're doing this model exactly if it's going to be that value, if it's going to be 20% higher, 20% lower. So we always carry out this one-way sensitivity analysis, and this generates something called a tornado diagram. It shows you which assumptions are the biggest drivers of cost effectiveness and hence potential pricing. So here's an example of a tornado diagram. So I didn't find my cursor again. Where's it gone? There it is. So this middle line going through the tornado, this represents the base case ISA. So you know on the screenshot I showed you beforehand, on the right hand side they were the sort of the base input value. So this represents the ISA from those values, which I think was around 91,000 if I recall. And then what you do is for each of your input parameters, so for example here we've got the health state costs associated with this GMFCS5 health state, and you vary that cost between an upper and lower bound, and that then shows you what the impact on the ISA is when you vary, and you keep everything else the same. So that gives you an idea of, well, if my costs for that GMFCS5 increase by 20% or decrease by 20%, how does that impact my ISA? And of course, the ones with the widest bars, which are the ones at the top, are the ones which are the biggest drivers of value. And they're obviously the things which are the most important. If you haven't got good evidence for those values at the moment, well, they're the values that you should be targeting in terms of trying to get more data and trying to look at observational studies. So you might want to make sure then that you're including those values in a future trial protocol, or they could be the subject of a separate real-world evidence study. So this is a case study which we did work on, which was for a gene therapy in dry AMD. It was a preclinical asset, and obviously it was a one-off treatment because it was a gene therapy. There was quite a broad range of target populations. So just on the right-hand side, these are just fundus images of an eye and just showing you the various different stages of this condition. So normal eye on the top left, you can have intermediate AMD, and then there was advanced AMD choroidal neovascularization, and then advanced AMD, different subtype geographic atrophy. So the client had a number of key questions and decisions to make. So which stage of AMD should they be targeting? What are the real drivers of value in this indication? Is it effect on vision loss? Is it safety? How do you measure those? How would you measure the visual? Is it visual acuity? Is it some other measure? Preventing progression? And how do you quantify these over what period? And obviously, the stage of the disease that you target, that affects both the potential price achievable and the potential number of patients. So one logically feeds the other. So in the little pie chart at the bottom, you can see that the vast proportion of the AMD patients were early stage, and then tiny little pieces, later on. So the two interact with each other. So obviously, you start off with a landscape assessment, and then we built an early economic model that was constructed to highlight any of these uncertainties in the demonstration of value, and then we built this alongside a forecast model. And basically, both of these helped inform a target product profile. It's like an iterative process. So you do your early economic model, you sort of find out the price, you feed that into your forecast model, and then eventually you sort of find the combination of things that lead you to the best target profile. And the three are interlinked with each other. So this is just a sort of an overview of the early health economic model structure. There were sort of 6 main levers, and these had multiple inputs and multiple scenarios where possible. And this model was used to develop pricing options based on a particular ISA, willingness to pay threshold. So on the top left, we've got the treated population. So we had a particular age distribution, a minimum treated age and a maximum treated age. And then the proportion of patients in different disease states that you were going to treat. You don't have to treat all your patients in one phase of disease. You might have a distribution so that you can enter that. There's some analytical inputs, the time horizon, which I spoke about earlier on, discount rate. So just as in accountancy, outcomes and costs in health economic models are discounted the further out you go from year 0. The AMD characteristics, so you had to input your progression rates from the different phases of diseases, but also the visual acuity decline rates per stage of disease and by age. Bottom left, your healthcare resource costs incurred by AMD stage, and then your quality of life. So there's a relationship between visual acuity and quality of life, obviously. And then the TPP characteristics. So what's going to be the impact of my treatment on the disease natural history? So that's the impact on progression, AMD stage, the impact on the rates of visual acuity decline, the duration of the treatment effect. This is often a big question when you have gene therapies. You think, oh, it's a one-off treatment, it's going to last forever. That's not true. You do get patients who receive gene therapies and they then may relapse. You have to capture that in your economic model because it has a huge impact on value. And then obviously the price. And in terms of the outputs, it generates, it's another metric called net monetary benefit. It's similar to the ISA. It's another just way of measuring value, and a positive net monetary benefit indicates cost-effective products, or anything which is above 0 is cost-effective at a particular willingness to pay threshold. And then the other thing it output was just obviously, well, what progression rate, what reduction in progression rate then generates a viable TPP. So these are just some screenshots. As I said, this was a, sorry.
Before you move on, can you go back? I just had a question. So as you look at it, positive net monetary benefits, would that be similar? It's like you're using a time horizon discount rate. What would the range of discount rates be to look at that NME? I'm thinking of it, I come from more of a business development background.
Right.
So I'm trying to understand how those two come together.
Yes. So I guess when you're doing a valuation, your WACC rate is often different per company, isn't it? But in health economics, it's normally determined by the group to which you're submitting. So if you go onto the ISA group website, it's 3%. So they normally, every health technology assessment body has got something called a reference case. So whether you go to NICE, whether you go to HAS in France, whether you go to ISA, somewhere you'll find details of what this reference case is, and this tells you what their preferred analytical inputs are. So 3% for ISA group. So yeah, that's not an area which is normally uncertain. There are some situations where there's a lower discount rate. So for example, NICE sometimes accepts a 1.5%, and that's obviously hugely advantageous if you have a one-off therapy. Because the disadvantage often is you've got hugely expensive upfront costs, but a lot of the benefits might come very, very far down the line, particularly if you've got a chronic disease where you've got a build-up of comorbidities over time. Those outcome benefits will be very heavily discounted, whereas if you have a lower discount rate, it won't affect your drug costs because it's an upfront therapy, but a lot of those outcomes down the line will be less discounted, so that generates higher QALYs. So some HTA bodies do give one-off therapies that benefit, or curative therapies that type of benefit. Okay, so as I said, so this is just some actual screenshots from this particular tool that was done in R-Shiny. So you can see down below the sort of coloured boxes are these health states that I mentioned earlier on. So we've got in early/intermediate AMD, we've got the choroidal neovascularization stage, and then the geographic atrophy stage. And the first thing you do in the model is you try and input all your values to do with the natural disease progression. So this is, forget about the treatment effect of your product, what happens for these patients without any type of disease modifying treatment. So here you can see you can enter, these are called transition probabilities if you look at the little ones, and this is basically the rate at which patients move from one stage to the other. Then the second thing you can put in is, if you, within each particular health state, there might be different rates of change in visual acuity over time. So it might be that the more progressed a disease is, you might find that progression and that loss in visual acuity happens faster. So you can enter these different rates of reduction in visual acuity by different stages of AMD. And then the final bit is just entering the relative prevalence of these different stages in the model, because as I said, when you start a cohort of patients on treatment, they might be distributed between these different stages, and it can often be quite interesting to look at, well, if I treat earlier stage patients, is their cost effectiveness much better than if my cohort's more skewed towards later stage patients? So that's what that slider does. Then the second bit, we've done the natural disease progression. The second bit is then looking at, is basically your target product profile. What is going to be the treatment effect of my drug? So the first bit obviously was a gene therapy. We had to think about, well, what was the treatment effect duration? At what point might patients then fall back to natural history progression rates? The second bit was the relative risk of progression. So how will my product affect the speed at which patients move from the early stage AMD to the intermediate to the late stage AMD? And then the final bit is then, well, how will my product then affect the rate of visual acuity decline for each of these different AMD health states? And once you've done that, what you can do is you can generate, here's quite a nice heat map between price and treatment effect on progression. And obviously, logically, as your product price gets higher, the likelihood of being cost effective is going to be lower, and obviously then also as your treatment effect, your reduction in those progression rates increases, you're then much more likely to be more cost effective. So this is obviously just a two-way heat map. We don't just deal with two-way analyses, it is possible to do multi-way scenario analyses. So all those different input boxes that I showed you before, those can be automated such that you could have a table, and I'm going to have value 1, value 2, value 3. For each of those different parameters in the model, I'm going to look at every single combination that I think might happen. It's a little bit more complicated in R-Shiny model, but in Excel it's really easy. You just set up a table with all the switches, you have a macro, you press the button, And then it shows you, well, what's the ISA with all these different combinations of input values. So that's essentially the cost effectiveness side. So the price. Obviously, when you're working on these types of assets, you want to also understand the impact on revenue and net present value. And this is where the forecast model comes in, which is probably where most of you guys tend to operate. So again, the forecast model was built in R Shiny. And the first bit here is just a drop down that allows you to either select a global view of your product, a global net present value, or inputs for individual countries. Because some clients might only be interested in the US, or they might just be interested in particular markets. There are some other slides coming up in case you can't. Is there something you're struggling to see, or you might see it better on some of the later slides? The second bit is just to put in the timing of the launch, which can be adjusted using a slider. But then the actual launch year is set in another sheet, which is set by the development schedule, which I'll show you later on. Obviously, you'd put in your price per eye, critical. However, it may be that might just be one eye's treated. It might be that both eyes are treated. So we've worked with a company where there was a lot of hoo-ha about actually this product was actually being transferred into the other eye after injection. So that's also something that you want to look at, because then you might have payers saying, well, why should I treat both eyes if actually you're getting some impact of the product just through one injection? And then the other thing is a discount rate for the net present value calculation, which is obviously completely separate to the discount rate that you use in your cost effectiveness model. You can select the stage of AMD that's targeted, and this is obviously going to be aligned with the cost effectiveness model. You can select multiple stages, but obviously the two need to be aligned. Whatever price that you've come up with in your cost effectiveness model, those stages need to be aligned with what you put in your net present value model. And then finally, the eligible age range is selected, so you can choose, the minimum or maximum age of patients that you're going to treat. So this is the screen just looking at the development phases. So again, you're probably familiar with this. So the first step in this particular R Shiny model was to enter the anticipated cost of each development stage. Obviously, preclinical phase 1, phase 2, phase 3. Regulatory, and then any phase 4 work. You enter the preclinical start year. That sort of then defines everything that happens down the line. Enter the duration of each phase of development, and then entering obviously the probability that you think each phase is going to be successful and move on to the next. This is all very standard stuff, which I'm sure you've all seen, but you might have typically done it in Excel. Rather than R Shiny. And then you have your outputs. So on the top right here is that you've got various different choices. We normally would generate an epidemiology funnel to show how you get from your overall patient population to the particular patient population that you're targeting. It then obviously generates patient numbers from that. This particular screenshot is the revenue screenshot that shows you the anticipated revenues by year, and then obviously the all-important net present value. And then this is just another screenshot of an output, which is your epidemiology funnel, just to give you an example again of how that works. Got to the end now, so it's good timing. So just to summarize about the benefits, so almost all innovative medicines will generate some type of resource use cost savings, but of course, overall new medicines usually will increase the total costs for the payer, and this is particularly true where the current standard of care is very cheap. And a cost effectiveness analysis allows you to articulate and quantify both the costs and the health benefits of a new product and ascertain what likely price comprises value for money to a payer. And because the cost per quality is a universal metric, regardless of indication, It can be used to compare the value of different pharmaceutical assets in completely different pharmaceutical areas, therapeutic areas rather. It can then be used in combination with a forecast tool, including epidemiology and market share estimates, and it can facilitate an assessment of likely value of an asset in different indications. This then supports portfolio management and obviously go and no-go decision making. It can support in-licensing decisions, and the use of sensitivity and threshold analyses can determine what level of efficacy may be needed to command a particular price, which facilitates development of a target product profile and clinical trial protocol design. And obviously sensitivity analyses can also determine which of your outcomes are most critical and would require evidence either from clinical trials or real-world evidence studies to convince payers of value. Yeah, it's come to the end now.